Nucleotides are the building blocks for DNA and RNA. By the help of phosphodiester bond, two nucleotides attach with each other and create a strand of DNA or RNA. Hello friends, welcome to Biology Concern. I am Animesh, and in this video I am going to discuss about the nucleotides, the basic structural difference between DNA and RNA. So let's jump into the topic. Firstly, nucleotides are the building blocks of the DNA and RNA. Nucleotides are made up of three basic structures. One is triphosphate. So first thing is triphosphate. So the structure of triphosphate is so these are the triphosphate. Okay. Second thing, a pentose sugar. Basically, this is a 5-carbon sugar. So if I draw the structure here, so this is the structure of pentose sugar. Okay, so here we have got this kind of structure, and this is the beta furanose. As number 1 carbon have the OH group upper side, so it is in beta configuration, right? So this is beta furanose. So this is the pentose sugar, and ultimate third, a nitrogenous base. Nitrogenous base, whether it is purine or pyrimidine. Okay, so these are the three things that make up a total nucleotide. I am already write down here the structure of purines and pyrimidines. So in case of purines, these are adenine and guanines. So these are the nitrogenous bases, and this is the structural conformation: 1 2 3 4 5 6 7 8 9. So in case of purines, these are 9 member. So these are the 9 members, and double ring present here, double ring present here. So this is the ring 1 and this is the ring 2. So double ring present here. And in case of pyrimidines, these are the 6 member. So these are the 6 members, and single ring present here. So single ring present here. So this is a 6 member, 1 2 3 4 5 6, and this is a single ring present here. In case of pyrimidines, in case of DNA, we have observed the cytosine and thymine, but in case of RNA, uracil replace the thymine. Okay, so in case the RNA, uracil replace the thymine. So these are the basic compositions of purines and pyrimidines. So if we put together all three of them, so what does the structure look like? So the structure going to be look like that. So these are the triphosphate. Okay, and here this OH, this OH and this OH, they are interacting with itself and releasing a H2O molecule or water molecule. So we have got this kind of structure. So this kind of structure, okay. And the thick bond or thick line means it is towards us, and the thin lines means it is opposite to us, because it is a 3D structure and we draw it in a 2D plane, okay. And after that, this H also get replaced with the particular OH group and releases water molecules, and it actually make up, so this actually makes up, okay. In this case, this is a cytosine. So we have draw the structure of nucleotide, the basic unit or basic unit building block for DNA and RNA. So what is the main structural difference between DNA and RNA? Here you observe, this is the nitrogenous base, this is the pentose sugar, so this is the pentose sugar, and this is the triphosphate, so triphosphate. So all together they are makes a nucleotide structure. But this is the structure of NTP, that is nucleotide triphosphate, or NTP is observed in case of RNA. So what is the difference between DNA and RNA? So in this case, in the DNA, we have got the full form of DNA is deoxyribose nucleic acid, okay. So in this case we have got a deoxy means here elimination of one oxygen occurs from the sugar, okay. So this is the sugar structure, and 1, this is 2, this is 3, this is 4, and this is 5. So these are the 5 carbons here. So in this case, here OH present and here also OH present. So this is the 2' region and this is the 3' region. Prime indicates the number of carbon of the sugar molecule, okay. So prime basically indicates the number of carbon molecule of the sugar, okay. Because in nucleotide triphosphate, in NTP or DNTPs, here carbon is also present in case of nitrogenous bases and carbon is also present in case of sugars. So how can we differentiate these carbon molecules of nitrogenous bases from these of the sugar bases? So we add a prime just of the number of the sugar. So 2' means it is indicates the carbon number of the sugar, so it is indicate the carbon number of sugar, okay. And 3' also, prime indicates the carbon number of the sugar. So this is the basic structure of nucleotide, and how we distinguish the carbon number from the sugar and the nitrogenous base, right. So here, so major difference DNA and RNA. So in this case, from here, if this structure is reduced, okay, if this structure is reduced and ultimately produces this kind of structure, so in here you observed, so in this case if you observe closely that we have got a hydrogen ion in case of hydroxyl ion. So the deoxy stands for elimination of oxygen, so deoxy stands for elimination of oxygen, okay. So in this case H plus replace OH minus, right? So in this case H plus replace OH minus and 2' position of that particular sugar. So if that is reduced by from OH to H, this is known as deoxyribose sugar. Okay, so deoxyribose sugar. Clear? So why we called it deoxyribose sugar? Because it lacks of oxygen. So it lacks of a oxygen. Okay, so it is now known as deoxyribose sugar. So in this case the NTP is now being called as DNTP. Okay, as because the particular number of the carbon of the sugar is reduced, and here in OH position H plus should join, and we observe the hydrogen in this case. Okay, so this is the basic or major difference from DNA and RNA. So nothing has been changed, but a single OH has been replaced by H plus ion, and this is denotes the DNTPs, that is deoxyribonucleotide triphosphates. And we have got DATP, DCTP, DGTP, and also DTTP. But in this case nucleotide triphosphates, we've got ATP, adenosine triphosphate, and in this case in the DNA deoxyribose adenosine triphosphate. So here in DNA deoxyribose adenosine triphosphate, deoxyribose cytosine triphosphate, deoxyribose guanosine triphosphate, and deoxyribose thymine triphosphate. But in this case adenine triphosphate, guanosine triphosphate, okay, cytosine triphosphate, and uracil triphosphate. As in this case no reduction is occurred, so no reduction is found in this case. So this is known as NTPs and this is known as DNTPs, right? So this is the basic difference between the DNA and RNA in structural view, and this is the structure of a nucleotide itself, where triphosphate, pentose sugar, and nitrogenous base play a crucial role to build up the whole nucleotide itself. I think this video is helpful for you to understand the basic structural difference between DNA and RNA and the composition or structure of a nucleotide. If you like that video, please hit the like button. Don't forget to subscribe and share. Thanks for watching.